Welcome guys. Let us make the topic of enzymes to be easy in biology paper 2. Right, so we are going to look at different questions from different past papers. Now we are going to begin with this question. So we have figure 2.1 shows the effect of pH on the rate of enzyme catalyzed reaction K and M. So from our diagram figure 2.1 we have two reactions denoted by letter K and letter M. Right, now if you look at the x-axis, we have one of the factors that affects the rate of chemical reaction, which is the pH. Now the pH, we can see the numbers are starting from 0 up to 14. So if we bring in science, which is chemistry, we know that 0 up to 6, this indicates the medium of acidity. So which tells us that reaction K occurs in acidic medium. Then we have this one from 8 up to 14. This is under the basic medium, which is a base. Then 7 tells us the neutral, the solution is neither acidic nor basic. So this is what we are supposed to know. So we are going to use now the pH and in line with the alimentary canal. So we have a bit of digestion, then plus the enzymes. Now let's have a look at question 1. So question 1, we are told what temp is given to pH at J and L. So let us identify J and L. So J is the highest point. So it is the highest point of our given curve. So this is our J. Then L is also the highest point. So what is the highest point or the peak of a given curve? So this is what we call optimum. So depending on the factor that you have under the x-axis, so for this one I'm going to say optimum pH, since we are dealing with the pH. Then we can now move on to question B2, where we are told to explain why the rate of reactions K and M goes down after point J and L respectively. So now after the optimum pH, why were the two reactions went down? So that's what we want to look at here. So we need now to bring in the concept of denaturing. So after the optimum, the enzymes becomes denatured, or they are denatured. So now what is to denature? What is to be denatured? So the denaturing of enzyme simply means that it has changed its original shape. And because of the changing of original shape, now the enzyme cannot fit on the substrate. So whenever you hear denature, we're not looking at where like destroyed. No, the denaturing simply means the enzymes have changed their original, original shape in such a way that they cannot fit on the, the substrate cannot fit on the enzymes. For example, let's say we have, this is the shape of our enzyme. Then we have the substrate with the shape like this one. So this is the normal because it can fit here. So what happens if the shape of this particular enzyme changes, like it becomes like a circle? So we know that this now cannot fit. So therefore, this enzyme has changed its original shape, and we can say it is denatured. So now we can say because the two reactions K and M so went past optimum pH, which caused the enzymes to be denatured. So this is what we can say. Now let's move on to the next question. So the next question is, we have question C. Name the regions of the alimentary canal where reactions K and M take place. So K, what did we say about K? We said K is under acidity, because we can see we have numbers from 0 up to 6. So this is acidity. So if you look at the alimentary canal, region where the medium is acidic is the stomach. So K is taking place in the stomach. So where is this acidic medium characteristic coming from? The presence of hydrochloric acid produced by the lining of the stomach. And I'm sure as long as you've studied the digestion, you may have come across HCl, which has so many functions. Then reaction M is under basic, which can be the mouth, because the saliva has a slight of basic nature. So we move on to question D1. Suggest one enzyme which can catalyze reaction M. So since we have chosen M to be in region of the mouth, then the enzyme that we can look at here is salivary amylase. And one thing that you need to know about most of these enzymes, their suffix is ASE. They end in ASE. Then we have Roman number 2. State one food nutrient which can be catalyzed in reaction K. So reaction K is where we are looking at the stomach, which has the acidic medium. So in the stomach, the food nutrient or the digestion that starts in the stomach is for protein. So this is what you need to know. Then we have question E, the last one for this question. State 2 factors other than pH which affect enzyme activity. So we have temperature. So temperature can make the enzymes to be inactive, or the temperature can make the enzymes to be denatured. So it depends on the amount of temperature. For example, the average normal body temperature of a human being is about 37 degrees Celsius. Above, for example, if you look at 38, this is abnormal. Then if we have 35, this is also not normal. Because 35, it simply means that the temperature is too low. Therefore, the enzymes will be inactive. They'll be there, but they have no energy to be active. Then 38, this is now too much of the temperature. Therefore, the enzymes can be denatured. And whenever the enzymes are denatured, then there is no life for that individual. Then apart from that, we can look at concentration. So it can be concentration of substrate or concentration of the enzymes. Then the third one, we have inhibitors. So inhibitors, we have 2 types of inhibitors. They are what we call competitive inhibitors and non-competitive inhibitors. So the only difference is that the competitive, they compete with the substrate. Instead of the substrate fitting on the active site, the inhibitors now will fit on the active site, preventing the substrate from combining with the actual enzymes. Then the non-competitive enzymes, they just make the shape of an enzyme to change. So you need to remember that. Right, so now we can move on to question 2. Figure 2.0 shows a graph showing the effect of, this time we are dealing with temperature, on an enzyme catalyzed reaction. Right, so from the given graph, we can see temperature in degrees Celsius in the x-axis. Now, the first question, question A, Roman number 1, is what is an enzyme? So, the enzyme is a substance. Is a substance produced by a living organism. Which act as a catalyst. So with me, I have issues with that definition of enzyme being the biological catalyst. In actual sense, enzymes are not catalysts. They just act as, for example, we have the president. If the president leaves the country, he leaves someone to act as a president. By natural sense, that person is not the president. So that is what you need to know about enzymes. So according to new research, this is the best definition. It's just a substance produced by living organisms which act as a catalyst. So let's have a look at Roman number 2. So Roman number 2, what is the optimum temperature of the enzyme reaction shown in the graph in figure 2.0? So to answer this question, we need to look at where our optimum is. So remember, optimum is this point where the graph has its peak. So I'm going to follow the dotted line. In case we didn't have this, you are supposed to draw a dotted line from this optimum. So remember, this is where our optimum is. So I'm going to draw a dotted line. So the point where this dotted line will cut the x-axis represents the number for this optimum temperature. So since here we have 35, this can be 37. So it depends on your estimations. We are not saying questions like this may have different answers. One may answer this one as maybe 36, 36.5, 37. All these will be considered to be correct, as long as the range is making sense. So for me, I can see that one as 37 degrees Celsius. Then we move on to question B1. At what temperature in figure 2.0 is the enzyme completely denatured? So how do you tell that the enzyme has completely denatured? So this is when the reaction has stopped. So the reaction has stopped completely because of no enzymes to act as a catalyst. Now, using the graph, the line, you need to look at this line. The line where it is just touching the x-axis, that point is where, or that shows where the reaction is completely down. So if you look at this is 55. So this can be 57 from what I can see. So say 57 degrees Celsius. Then reason we can say because the reaction completely stopped. Right, so we move on to question C. So C1, explain why it is important to measure the body temperature when a person is sick. So when we take a sick person to the clinic or hospital, why do they measure body temperature? So here we don't need answers like to see if the person is sick. That's not the answer. That's not biologically correct. What we need is why is that important. So the reason why they are checking the temperature, they just want to see if the temperature at that point is still favorable for the enzymes. Because as we move on, as we are live, we need enzymes for us to survive. We need enzymes so that the metabolic reactions could still take place. So if the temperature goes beyond the normal, then the increase in temperature may result in damaging the enzymes. Then if the enzymes are denatured, the reaction will stop. When the reaction stops, then there's no life. So the enzymes that we have in our body plays important role in chemical reaction. So they help us to be alive. That's the point. So here we can just say to check if the body temperature is still favorable for the enzyme's activity. So that's what they just want to see, if that temperature is still favorable. Then propose 2 ways the body temperature of a sick person can be brought down to normal. So drinking a lot of fluid. Then number 2 can be by dipping the sick person in cold water. So these are questions like its application. You apply now. What you've learned now, apply. Right, so we can go to question 3. Figure 2.0 shows a protein molecule undergoing digestion in the alimentary canal of a human body. So the molecule that we are dealing with is a protein. And if you look at organic chemistry, a protein is a polymer. Since it's a polymer, therefore it is a long chain molecule, as we can see from the diagram. Then we have this reaction K. So reaction K helped the long chain protein to be broken into 2 parts. So we can see we have 2 here, 2 here, then 2 here, 2 here. So reaction K is just ending at the product of 2, 2. Then reaction M is now giving us single parts, we can see. So reaction M is just the completion of that digestion of what? Proteins, right? Now from this question A, Roman number 1, identify the products labeled L. So when a protein changes into a new product, so what product is that? Before it gives us the end products of amino acids. So we have what we call peptide. So we have peptide. Now, if you look at this peptide, there are 2, 2. We have 1, 2, 1, 2. Since there are 2, 2, therefore you need to add the prefix di. So this becomes the dipeptide. If they were 3, tripeptide. We even have polypeptide if there are many. So it depends on what you have. So 2, di, 3, tri, more than 3. You can just say poly, because poly means many. So we have dipeptide. Then we can move on to question Roman number 2. Suggest a region in the alimentary canal where reaction K takes place. So reaction K is where we are looking at the protein changes into the peptide. So this is just the region where the digestion of proteins begins. So it begins in the stomach. So that's our answer. So this is the stomach. As simple as that. Then let's look at the B. Reaction M completes the digestion of proteins. Which enzyme catalyzes reaction M? So we are looking at we have protein. So protein broken down into peptide. Then the peptide broken down into amino acids. So the amino acids are the end products. So the question is, which enzyme or enzymes are responsible to change the peptide into amino acids? So among of those enzymes, you just have to get the name peptide. So you just have to say peptide, then you end with ASA. So you just add the ASA. So whatever that you have here, you remove A, then you end with ASA. So peptidase is the enzyme. So what is pH at which reaction M would be catalyzed by the enzymes identified in B1 above? So the reaction M would be basic. This is now in the small intestines. So this would be basic. Then Roman number 3, suggest where the enzymes named in B1 above is produced. So it can be, so suggest where it can be pancreas, because pancreas has a pancreatic juice that has enzymes that completes, or we can say lining of the small intestines. And if you look at these small intestines, we are looking at duodenum, because the completion of mostly for proteins and absorption occurs in the duodenum. Duodenum is the first part of the small intestines. Then we have question 4. Identify the products labeled N. So the products labeled N are the end products. So these are now the amino acids. So you just have to remember that amino acids are the end products of protein digestion. Then describe 3 adaptations which makes the ileum suitable for, should be not digestion. I'm sure there was an error for absorption, because ileum is known for absorption. So absorption, number 1, we can say the ileum is long to increase the surface area. The surface area for absorption. Then it has thin epithelium. So the epithelium is thin, which now makes it easy for digested food to diffuse into the bloodstream. Then the ileum is supplied with blood vessels, which now transports the food values to the liver for inspection. Right, so now we can move on to the last question for this video. Figure 2 shows the stages involved in an enzyme catalyzed reaction inside cells of a living body. So we have part G, H, and I, stage 1, stage 2, stage 3. Right, so for the first question is we are told to from stage 1. So here, in figure 2, identify the enzyme. So now to fully understand how to identify an enzyme, you need also to focus on the final stage. So if you look at the final stage, you need to look at which substance or substances did not change their shape. So if you look at this, the structure of the eye remains the same. But for G and H, they were combined. So this structure that never changed its shape is an enzyme. So we just say I. So just say I. Then substrate will be G and H. Right, so we can move on to question B1. So which stage shows the enzyme substrate complex? So enzyme substrate complex, it's a stage where the enzyme and the substrates now have bonded together. They like attach to each other. So that's the stage that we are looking at. So if you look at the 3 stages, this stage shows that the enzyme and the like now have bonded together. So that is stage 2. We have stage 2. Then Roman number 2, label the active site of the enzyme on figure 2. So the active site is just the point where the enzymes attach them, the substrate attach themselves to an enzyme. So this is an active site. Also, this is an active site. So as long as it's a point where these substrates meets with the enzyme, that is the point that we were looking at. So we can move on now to question C1. What type of metabolic reaction is catalyzed by the enzyme in figure? So if you look at the way in stage 1, the substrate, the 2 substrates are individually, but if you look at the stage 3, the 2 substrates have combined. So we are looking at the reaction in which you have 2 or more monomers that combines to give you 1. So this is what we call anabolic. Remember, anabolic has to do with the building up. So it's anabolic reaction. Then if it was you have something that is combined, then after a certain reaction, it breaks into, let's say, 2 products. Then this could have been catabolic. So catabolic is a breaking down, anabolic is the building up. Now, remember to name a region in the human body where such a metabolic reaction in C1 above can occur. So, from the human body, where would we expect to have the process of anabolic? So, number 1, liver and kidneys. Now, state the general term used to refer to enzymes that work inside the cells. So those that works inside the cells are called intracellular enzymes. Then for those that works outside are called extracellular cellular enzymes. Then describe two industrial application of enzymes. So number one, enzymes are used. So enzymes are used in number one, baking. Right, so let's not in baking, but let's make it in brewing, when making alcohol. So brewing enzymes are used in brewing. Then also enzymes are used when making dairy products. Even in making soaps, we have enzymes. That's why on some of the covers of soaps, we may have seen a multi-purpose enzyme. So this is how we can answer questions on this. So for more, come for online classes. So that's it for this video. Thanks for watching.